Awesome. Thank you so much, Lida and Aton. That was a wonderful talk. Next up, we have for Discovery Lab, Shelby Spindler is going to be giving our talk, and her talk is entitled Drosophila melanogaster as a model for identifying potential chemotherapeutics.
Okay, thank you, Gretchen, for introducing me. So I'm just going to give you a quick outline of our class, why we're even talking about cancer, why we need new treatments, some experimental design and how we designed our class this semester, what methods we used. I'll highlight a couple independent projects that we thought were really cool this semester, and then we will wrap it up. So why are we even studying cancer? Why is this a problem? It's been shown that about half of all Americans are going to develop cancer in a lifetime. I'm sure every single one of you in the audience knows at least one person who's been affected by cancer. And the treatment outcomes are not really improving. So down here on the x-axis, we see the year, and here on the y-axis is the death rate per 100,000 individuals. And if we look down at these down here, since about the 70s, even though we've had a lot of technological advances and treatment advances since then, the outcomes are not really improving. And if this many people are getting cancer and over this many years, the outcomes are not improving, then that's a really big problem. There's a lot of different ways cancer can develop. A big way is from mutations. So we have proto-oncogenes and tumor suppressor genes. These are normal everyday genes. They are good to have, but when they're mutated, that's when it can become a problem. So over here, this is a great kind of analogy using a car. So up here we have our normal genes. They regulate cell growth and they are all good, but then if we have a mutation in one of the gene, let's say the proto-oncogene, then it becomes an active oncogene after that. Then that means we no longer have the acceleration, and let's say we have a second mutation that occurs, and in this case, that's going to lead to cancer. So now we don't have any brakes. The tumor suppressor gene is the one that's mutated. And if we don't have any brakes on division, then we're just going to accelerate at full speed. The cancer is just going to rapidly divide, and that's a big reason it leads to cancer, and that's a big problem. There's also a lot of challenges in treating tumors like influx and efflux pumps. So influx pumps, basically, they pump in the chemo. So let's say you're using chemo, the tumor drug, it's going to pump it in, and efflux pumps are what's going to pump the chemo out of the cell. So if we downregulated influx pumps, then that chemo couldn't get in as easily to kill the tumor. And if we upregulated efflux pumps, then everything that does get in, the tumor cell is just going to pump out. So that's a challenge to treating it. Survival rates are really low too for cancer. So we look at this graph over here, just some major cancer treatment centers in the US, like Mayo, this blue one, I'm sure most of you have heard of. We look at the survival rate, which is going to be over here. We all start at 100%, and after about 10 years and on, we see that the survival rate is about 25%, and it's similar for most of them. And that's awful. It's really harmful for the patient, especially with chemotherapy as a treatment, because some of them can be so toxic. There's a very fine line between killing the patient or killing the tumor. And then there's a lot of drug resistance too. This next slide shows a great depiction of that. So we have this red cell that's going to be resistant right here in this whole tumor. And if we treat it with a traditional cancer therapy, then all of those other cells could die off, but this red cell that's resistant could still be alive, and that could lead to a whole new tumor, and that's not going to be resistant to that cancer treatment anymore. But if we use a combination therapy, which is what we're doing in drug discovery, we're trying to look at the synergistic effects of chemotherapy and radiation. So what that means is that we're taking the chemo and the radiation, we're combining them together, and we hope that those together can produce a higher effect than either of them would alone. So you can see if we use combination therapies, we have a lot better chance of killing even the resistant tumor cells, and then we wouldn't have that relapse anymore, which is what our goal is. So a little bit of how we designed our experiments. So we used Drosophila melanogaster as our model organism for tumors. So these are just your wild type fruit flies. They themselves don't have any mutations. They don't have any tumors in them. But we use them as our model organism for tumor, this third instar larvae right here, because they have really rapidly dividing cells that mimic tumors. They're also really responsive to the chemotherapy and the radiation that we give them, which can give us a good representation of how the tumor is going to respond. And because we're doing these testings over and over again, they have a short life cycle and they're inexpensive, which is good for that. So like I said, we collect this third instar larvae. So first we're gonna isolate these embryos, and we isolate those from population cages. All the adult flies will go and lay their eggs in this grape juice plate. We let that incubate, and then we isolate this third instar larvae using sieves. So we have 3 different sieves of different sizes, and all of the third instar larvae are the same size. So those get caught in the middle of the sieves. And we will take that and radiate them with 2,500 rad, which is just a crazy amount of radiation. And then we let them incubate a little more before we used all of our compounds, which were different for everyone. We diluted those series into food. And then we put the third instar larvae into the food and quantified their survival after about 14 days. And how we quantified survival was the larvae would climb up the walls of the vials and they would lay pupal cases. So remember, since Drosophila melanogaster is our model organism for the tumor, we are expecting it to kill the tumor, so we are looking that all of the flies died. So these empty ones means that the flies climbed out of their pupal case, and that means that they are alive. So we're looking for all of these ones that weren't fully closed, which means that they died in their pupal case. So how we identified which compounds that the students chose were potential hits was through this graph here. Kind of explains it. So this is just our negative control, which is DMSO, and we expect a 50% survival when treated with it with radiation. And you can see our mean right here, this red line, we have about 50%, which is great. Anything below 2 standard deviations of the mean is statistically unusual. So that's going to be what we consider our hits, of what our potential chemotherapies are. So anything below this line is what we're looking for, the potential chemotherapies. So a couple independent projects. Dr. Harvey gave us this huge list at the beginning of the semester of places we could choose our compounds from. So we had some from the FDA approved drug screen. We also had some compound library hits up here that we didn't know their effects of. And a lot of them came from this previous student selections one down here, which is really great because our goal at the end of this is to try and find a chemotherapy that works. And if we can use student selections that have been potential hits in the past, and we can continue experimenting on it, then we can get closer to that goal. So that's where a lot of them came from. So one really cool one was Androgylide. This was from Acacia, Corbin, and Shreya. So this is from the Andrographis paniculata plant that's native to India. Research has found that it promotes apoptosis, which we know is cell death. That would be great. And they got this from a student choice in a previous semester. So we can see our positive control over here is colchicine. That's an already known chemotherapy. So we expect our percent survival of the flies to be 0 with that, since it's an already tested and known chemotherapy. Our negative control, which is the 50%, which is what we expect. And then here's that minus 2 standard deviations of the line. And anything below that is what we're looking for, potential chemotherapeutics. And you can see all 4 of their doses right here were under that 2 standard deviations, which means they were hits, which is great. That's what we're looking for, their potential chemotherapies. So this is really interesting because androglyphylide is a natural compound, and that can show that some chemotherapies are super toxic, but if you can find more natural compounds, then it can help not harm the patient as much. So in a similar study, this grapeseed extract from an actual grapeseed was studied by Jack, Mayu, Dylan, and Haley. And same thing, this is the minus 2 standard deviations. And we can see over here is their dosage series of their compound. And they also had hits, which was fantastic. So these results kind of emphasize that the promise of some natural compounds as potential chemotherapies that, like I said earlier, they don't have to be as harmful to the patient, but they can still be just as effective in killing the tumor. So on a different kind of note, this is Valproic Acid. This was by Kenya, Rose, and Mariam. It's already an FDA-approved drug to help treat bipolar disorder, but we tested it as a potential chemotherapy. It can inhibit mechanisms of DNA replication, which is great because we all know, like I said earlier, with not having the break, replication just keeps occurring. So we can see, again, this red line is the 2 standard deviations of the mean, and they also had some hits. So this was also from a previous semester. This kind of shows that there's a lot of different places you can get chemotherapies from, and some can be natural, like the ones before, some cannot be as natural. But if we can get this huge list of data and huge list of compounds and kind of narrow it down, then we can get towards that goal. Which brings me to my conclusion. So we found that radiation effectively reduced survival rates by 50%. And that's the negative control DMSO. That's what we expected. So we tested 27 compounds as a class total. And of those 27 compounds, 20 of them were hits, which is just amazing. That means 20 of them are potential chemotherapies, and we can further test those. So for the future, we want to test those 20 hits in a new semester. And we're looking at trying to get towards the goal of finding actual chemotherapy. So we can see from this graph down here, it takes a long time and a lot of work and money and effort and everything into going a clinical trial. So we didn't start with 10,000 compounds, but if you start with a lot of compounds and your goal is to get one approved FDA compound, then we can take those compounds from last semesters that were already identified as hits. We can take ones that we identified this semester, and we can kind of start funneling in to get towards the goal of doing clinical trials. And then hopefully by the end of it, we will have an actual chemotherapy that will work with radiation to help improve cancer patient outcomes. Couple acknowledgements real quick. I'd like to thank Dr. Tintin Sue. She was the PI for this lab. Dr. Pamela Harvey was our instructor. Gretchen Hoyer and Kiana Roberts, our lab managers, and Ellie O'Brien, my TA, and all of the TAs. We couldn't have learned any of this or done any of this without you all. And also for the funding, Howard Hughes Medical Institute, Biological Sciences Initiative, and the Department of MCDB. So thank you all. Is there any questions?
I see one question in the chat, just kind of Jared being curious whether a bipolar drug as a potential drug as a chemotherapy. He says he wonders if there are people already taking the drug who have reduced rate of certain kinds of cancer. Maybe not something interesting.
So I'm not sure about that, but one thing, let me go back to it. That's cool about using an already approved FDA drug is that if our goal is to get to clinical trials, phase 1 is going to be testing the toxicity rate. So what dosage can we give them that's effective, that doesn't kill them? So even though this has been for bipolar, which doesn't have a lot to do with cancer, it's cool to use something that's already FDA approved since we already know that toxicity rate. We wouldn't even have to do that part of the trial.
Oh yeah, that's a huge advantage.
Yeah, it is. Thank you.
And I have a question. Is it known what's in grapeseed extract? I don't know if the grapeseed extract group is here. I know that that's not your specific research.
Yeah, personally, I don't know, but I know it's a natural compound. If they would like to chime in, they totally could. If not, that's all right too.
And then I got a question. How are these compounds chosen to be studied to begin with?
So let me go back to that. So Dr. Harvey will literally call up the FDA, like from the FDA approved drug screen, and she'll be like, give me the compounds that you've got. So a lot of these, the FDA have already tested themselves and they were hits. So we can use ones that they've already tested. We can use, these are also from the FDA, but we just aren't quite sure of what their effects are. So these ones right here and these ones we got from a list from Dr. Harvey calling them. And then we could choose these ones from just past semesters of what they've already done.
Any other questions?
I had one, and it might be like completely wrong, but since drugs are toxic, could there be like some short-term chemotherapies that long-term become carcinogens?
I'm not quite sure about that. I'm sure they could. That's kind of one of the hard things about treating cancer is because their drugs are so toxic. That's why we're trying to do treatment synergistically, because if we can use something that's not quite as toxic with a radiation that can kill the cancer without being as harmful to the patient, that's like the best outcome. And that's what we're trying to go for.
Perfect. And that's all the questions we have time for. I think there's a couple more in the chat, but if you have burning questions, you can always stay after or ask us later.